You wake up tomorrow. Let's say it's 6:47 in the morning, and the kitchen is quiet except for the sound of a knife splitting the skin of a single ripe avocado. You scoop half of it onto a plate, next to 2 eggs. In a few minutes you're going to eat it, and before you do, I want you to sit with one fact. If your breakfast this morning were nothing but toast and jam, your stomach would push that food into your small intestine in under an hour. Swap in that half an avocado. Same plate, same person, same morning. And your stomach holds onto the meal for closer to 2 or 3 hours. That is not a wellness slogan. That is a measurable difference in how long food physically sits inside you, and by the end of the next few minutes, you will know exactly which nerve carries that signal, which hormones get released along the way, and why your own gut contains something physiologists literally call a brake. This isn't a detox video. There's no toxin being flushed, no metabolism being boosted, no miracle molecule found in exactly one fruit from exactly one part of the world. What's actually happening is ordinary, well-mapped physiology. The same physiology that plays out every time you eat fat and fiber together, whether it comes from an avocado, a handful of olives, or a spoon of natural peanut butter. Once you understand the mechanism, you'll be able to explain it to somebody else, in plain words. Why a few ounces of green fat in the morning can quiet hunger for hours, why that has nothing to do with willpower or clean eating, and, just as important, which parts of this story are proven, which are modest, and which are still an honest, educated guess rather than settled fact. Let's start with what you can actually feel, because your body knows this before your brain does. If you've ever eaten a bowl of cereal or a plain bagel for breakfast, you know the pattern. You're satisfied for maybe an hour. Then, somewhere around 9:30 or 10, there's a pull. Not dramatic, just a steady tug toward the biscuit tin or the vending machine down the hall. Swap that same breakfast for one that includes real fat and fiber, and that mid-morning pull is quieter. Sometimes it doesn't show up at all. That difference isn't discipline, it's plumbing. Here's the part most people get wrong. They treat hunger like a fuel gauge, ticking down at a steady rate based purely on how many calories are left in the tank. It isn't that simple. Hunger is a decision your brain makes, moment to moment, based on chemical signals arriving from your gut. And those signals depend enormously on what kind of food is sitting in your stomach and small intestine, not just how much of it there is. So let's go inside and look at the tissue itself. An avocado is mostly water and fat, with considerably more fiber packed into it than people expect from something they'd casually call a fruit. That fat is overwhelmingly a molecule called oleic acid, the same monounsaturated fat that makes up most of olive oil. When you swallow a bite, your stomach's lower, muscular section, the antrum, starts grinding it through a set of powerful contractions, mixing it with acid and enzymes into a semi-liquid mixture called chyme. But fat is stubborn. It doesn't break apart the way starch or sugar does. It needs bile, made in your liver and stored in your gallbladder, to physically emulsify it, breaking big globules into smaller droplets, the same way dish soap breaks grease into tiny beads floating in water. And it needs an enzyme called pancreatic lipase, released from your pancreas into the small intestine, to actually cut those fat molecules apart into pieces small enough to absorb. How thoroughly you chew plays a small supporting role here too. Breaking the flesh into smaller pieces before it even reaches your stomach gives bile and lipase more surface area to work on. It's a minor factor next to the fat and fiber content itself, worth knowing, not worth obsessing over. Here's the key mechanical fact, and it's the hinge the rest of this video swings on. Your stomach wall contracts in a steady underlying rhythm, roughly 3 times every minute, whether or not there's fat in it. That rhythm barely changes. What changes is a valve at the stomach's exit called the pylorus, and whether it's willing to open with each of those contractions. That valve does not run on a fixed schedule. It responds to what's waiting on the other side. When the first stretch of small intestine, the duodenum, is already dealing with a heavy load of fat, it sends a signal back that tells the pylorus, in effect, not yet. The valve tightens. Emptying slows, even though the stomach is still contracting at the same steady pace it always does. A fatty, fibrous meal doesn't leave your stomach in one dramatic wave. It leaves in small, metered pulses, spread over a far longer window than a plate of plain carbohydrate ever would. This isn't guesswork dressed up as science. Back in the 1950s and 60s, a physiologist named John Hunt ran some of the first careful, direct studies measuring how quickly different meals actually leave the human stomach. Using test meals and repeated sampling to track the process in real time, fat consistently came out as the slowest macronutrient to clear, well behind sugar and starch. That basic finding has held up for 70 years of follow-up research since. Your stomach isn't a bucket that drains at a constant rate. It's a valve system that reads the composition of what's coming through and adjusts its own speed accordingly. Researchers today don't have to guess at any of this either. The standard way to measure it is a technique called gastric emptying scintigraphy. You eat a meal laced with a tiny, harmless radioactive tracer and a gamma camera positioned over your stomach tracts. In real time, how much of that tracer is still sitting there at 15 minutes, 30 minutes, an hour, 2 hours? It's how we know, directly rather than by inference, that a fattier meal can sit in the stomach meaningfully longer than a low-fat one of the same size and calorie count. There's a second, entirely separate break running alongside the hormonal one, and it comes from the fiber itself rather than the fat. A portion of that fiber is soluble, meaning it dissolves in stomach fluid and swells into a thick, viscous gel. Picture cornstarch stirred into water until it turns syrupy rather than watery. That thickened gel physically slows how easily stomach contents can squeeze past the pylorus, and further downstream, it slows how quickly glucose molecules can diffuse across the intestinal wall to be absorbed. This one has nothing to do with hormones or nerves. It's pure physics, sitting right alongside the chemistry, and it's one of the more consistently replicated findings in research on soluble fiber and blood sugar. Now, here's where it gets genuinely interesting, because the story doesn't stop at the stomach. It moves downstream, into chemistry. When fat and partially digested protein reach that first stretch of small intestine, specialized cells in the intestinal wall, called eye cells, release a hormone called cholecystokinin, or SEEK. Back in 1973, 3 researchers, Gibbs, Young, and Smith, ran an experiment that reshaped how scientists understood hunger. When they gave animals a dose of SEEK, the animals stopped eating on their own, well before their stomachs were anywhere near full. That was some of the first clear evidence that satiety isn't only a stretch signal from a swollen stomach. It's a chemical signal, triggered specifically by the presence of fat and protein moving through the gut. The hormone's own name is a small piece of first principles science hiding in plain sight. Cholecystokinin literally means gallbladder mover, chole for bile, cysto for sac, kinin for movement. Because decades before anyone understood its role in hunger, researchers had already noticed it, making the gallbladder squeeze and release bile the moment fat arrived in the gut. Its appetite-suppressing job was discovered later, layered onto a hormone whose name already described half of what it does. Here's the rehook, because this is exactly where most explanations quietly stop. Right before the best part. SEEK is only the first messenger. The story keeps traveling further down the tract, if fat makes it far enough. Past the duodenum, into the far end of the small intestine called the ileum, it triggers a second set of cells, called L cells, to release 2 more hormones, GLP-1 and PI. In 2002, a research team that included Rachel Batterham published a study in the journal Nature showing that PI, on its own, physically reduces how much people go on to eat at a later meal. And this is exactly where the mechanical break gets its actual scientific name. In 1984, a gastroenterologist named David Spiller published research describing this precise reflex: fat reaching the ileum, and the gut responding by slowing everything upstream of it, including how fast the stomach empties and how vigorously the intestine contracts. He called it the Ileal break, and the term has sat in physiology textbooks ever since. Picture your digestive tract as a road, and gastric emptying speed as a car's cruise control. Simple carbohydrate is like flooring the accelerator. Food surges through fast, blood sugar spikes fast, and hunger comes roaring back just as fast, because the tank drains just as fast. Fat and fiber, once they reach that far checkpoint in the ileum, tap the brake pedal. Not slam it, tap it. The car doesn't stop. It just covers the same road more slowly and more evenly for longer. None of this is happening in your stomach in isolation either. It's happening in your brain, in real time, while you're still sitting at that table. The signals I just described, stomach distension, seek, GLP-1, PI, don't only act locally. Many of them are picked up by the vagus nerve, the long nerve running from your gut straight up to your brainstem. Vagal fibers carry that information to a small structure called the nucleus tractus solitarius, sitting at the base of the brainstem, functioning like a relay station. From there, the signal passes up into a region buried deep in the hypothalamus called the arcuate nucleus, the part of your brain responsible for regulating hunger, temperature, and a handful of other basic survival functions. Most people picture nerves as carrying commands outward, from brain to body. The vagus nerve mostly runs the opposite direction. The majority of its fibers are sensory, carrying information up from the gut to the brain, not orders back down. In a very real, anatomical sense, your gut talks to your brain far more than your brain talks back to it. Inside the arcuate nucleus sit two competing populations of neurons. One set, called AGARP and NPI neurons, drives hunger. Call them the accelerator. The other, called PMC neurons, drives satiety. The brake, again, this time inside your skull rather than your gut. Ghrelin, released mainly by an empty stomach, activates the hunger neurons. C-key, GLP-1 and PI, arriving by way of the vagus nerve, dampen them down and switch on the satiety neurons instead. Your sense of, "I've had enough," isn't one clean switch flipping. It's the running scoreline of a tug-of-war, refereed largely by whatever is currently sitting in your gut. There's one more detail about GLP-1 worth knowing, because you've almost certainly heard its name before, just in a very different context. GLP-1 is the exact hormone that prescription medications like semaglutide are engineered to mimic. The drugs now widely used for diabetes and weight management, which activate GLP-1 receptors in a sustained, pharmacological way, around the clock. What your gut releases naturally after a fat and fiber breakfast is the very same molecule, but in a completely different magnitude and duration. A small, transient rise lasting a couple of hours compared to a steady, engineered activation lasting all week. Eating an avocado is not a gentle, natural version of that medication. It's the same lock, turned by a far quieter key for a far shorter time. I'm only naming this so the comparison doesn't get inflated into something it plainly isn't. GLP-1 earns its keep in a second way, too, through something called the incretin effect. Back in the 1980s, researchers, including the German diabetologist Michael Nock, showed that drinking a dose of glucose triggers a noticeably bigger release of insulin from the pancreas than delivering that exact same amount of glucose straight into a vein. Your gut, it turns out, sends a signal ahead of the sugar even finishing absorption, priming the pancreas to respond harder specifically because the sugar arrived by mouth. GLP-1 is one of the two main hormones responsible for that priming. It's a second job, layered onto the same molecule already tapping your ileal brake. One role for hunger, a separate role for how hard your pancreas responds to whatever glucose actually gets through. Now let's connect this to blood sugar, because slower gastric emptying doesn't just change hunger, it changes how fast glucose enters your bloodstream in the first place. If your breakfast includes any carbohydrate at all, the toast underneath that avocado, say, a stomach releasing its contents in small, metered pulses will also release that glucose more gradually into the small intestine, where it's absorbed. A slower glucose entry generally means a gentler insulin response, rather than a sharp spike followed by an equally sharp fall. That fall, that rebound, is a large part of what people actually experience as the mid-morning energy crash. This can even carry forward into your next meal, not just this one. Back in 1982, a research group at the University of Toronto led by David Jenkins described what they called the second meal effect. Eating a meal that produces a gentler blood sugar response doesn't just help in the moment, it measurably improves how your body handles blood sugar at the meal after that, hours later. A calmer breakfast can leave a quiet echo sitting underneath your lunch. And the story has one more stop before it's finished, because not all of that fiber gets absorbed up in the small intestine at all. Some of it travels all the way down to your colon, where resident gut bacteria ferment it into short-chain fatty acids, molecules like butyrate. Those short-chain fatty acids do two things worth knowing. They serve as a direct fuel source for the cells lining your colon, and they can prompt colonic L cells to release still more GLP-1 and pi hours after the meal itself is technically over. This slower, second wave of signaling is well documented in gut physiology generally, though exactly how much of it comes specifically from avocado fiber, as opposed to fiber in general, hasn't been measured meal by meal in the same detail as the stomach-level effects. I want to flag that honestly rather than smooth over it. So here's where all of this actually matters for you, at your age specifically, and not just as an abstract diagram. As we get older, insulin sensitivity tends to decline. One of the more consistent findings in aging research, including long-running work like the Baltimore Longitudinal Study of Aging, which has tracked metabolic changes in adults over decades and found this decline even independent of weight gain. Cells simply respond a little less efficiently to insulin's signal than they did at 30. That means the blood sugar swing following a fast-emptying, high-carbohydrate breakfast tends to be somewhat larger, and the crash that follows tends to be somewhat more noticeable. More fatigue, more fog, more of that pull toward a second coffee or a pastry by mid-morning. A breakfast that mechanically blunts the speed of glucose entry isn't a cure for any of that. But it is a small, real lever, working through the exact chain we just walked. Stomach, valve, hormone, nerve, brainstem. On a system that's carrying a slightly heavier load than it used to. There's a second piece that matters at this stage of life, too. Appetite regulation itself tends to grow a little less precise with age. Research on older adults has repeatedly pointed to blunted hunger and fullness signaling in general, and that can cut in either direction. Some people quietly undereat because their hunger cues fade, which raises real concerns around muscle loss and frailty. Others find themselves grazing more because their fullness cues arrive late or arrive weakly. A meal that reinforces satiety mechanically, through fat, fiber, and simple volume, rather than through willpower, hands your brain a stronger, clearer version of a signal it might otherwise be sending faintly. There's a further consequence worth knowing at this stage of life, since so many of us are managing at least one regular medication by now. The same delayed gastric emptying that blunts glucose absorption also delays how quickly anything else swallowed with that meal reaches the small intestine, where most drug absorption actually happens. This is an established general principle in pharmacology, not something unique to avocados. Taking a pill alongside a heavier, fattier meal can shift both the speed and, for some medications, the total amount absorbed, compared with taking it on an empty stomach. It's part of why a pharmacist's instruction to take something with food or on an empty stomach isn't a throwaway line. It's accounting for exactly this mechanism. I'm not suggesting you change how or when you take anything. That's a conversation for your own doctor or pharmacist, medication by medication. I only want you to see that the break we've been tracing doesn't stop at glucose. It touches nearly everything else riding through your gut at the same time. If any of this sounds familiar, that mid-morning crash after a carbohydrate-heavy breakfast, or the opposite, forgetting to feel hungry until you're already running on empty, tell me which one, in a comment, because it actually tells us something different about which side of that hunger and satiety tug-of-war tends to run quieter for you specifically. Now, before we go any further, I owe you the honest version of this story, because a good teacher tells you plainly what's proven and what isn't. The gastric emptying slowdown from fat is about as well established as digestive physiology gets. Decades of direct measurement, going back to Hunt's original work, replicated many times over since. The existence of SEEK, GLP-1, and pi, and their release in response to fat and protein in the gut, is basic, textbook endocrinology, not a matter of debate. The ileal brake reflex itself, as first described by Spiller, is a recognized, named physiological mechanism, not a theory waiting to be confirmed. Where the evidence gets thinner is in the size of the practical, real-world effect. And this is exactly the spot where marketing tends to sprint ahead of the science. In 2013, researchers at Loma Linda University, led by Joan Sabate, published a study in the Nutrition Journal in which overweight adults ate a standard meal with half an avocado added, compared against the same meal without it. People reported feeling more satisfied and less interested in eating for several hours afterward. That part held up cleanly. But when the researchers actually measured how much food those same people ate at a later test meal, the difference was much smaller and far less consistent. The subjective feeling of fullness didn't translate into a dramatically lower calorie intake down the line. That's an important, honest detail. The break is real. It's a gentle tap, not a slammed pedal. It nudges behavior. It does not override it. A few pieces of this story are also physically plausible without yet being directly nailed down, and I'd rather tell you that than pretend otherwise. The idea that this mechanism matters more specifically as insulin sensitivity declines with age is a reasonable inference from what we already know about aging metabolism in general. It hasn't been run as a dedicated avocado and aging clinical trial, isolating that exact variable in older adults. That gap is worth naming rather than quietly stepping over. It's also worth remembering where some of this evidence originally came from. Gibbs, Young, and Smith's original C-key experiment, for instance, was done in animals. The human satiety research that followed later confirmed a similar effect in people, but the very first demonstration of the principle wasn't in a person at all. That's normal, honest science, built up in layers over decades. It's just worth knowing which layer came first. And not every avocado delivers this identically. Riper fruit and different varieties carry somewhat different fat content, and the effect we've spent this whole video on depends on that fat and fiber staying bound up together inside actual plant tissue. What nutrition researchers call the food matrix. Pressed avocado oil, stripped of that fibrous structure, empties from the stomach and gets absorbed considerably faster than the same fat still locked inside the whole fruit cell walls. Everything we've traced tonight belongs to the whole avocado, spoon and all, not to the oil sold in a bottle beside it. One more thing worth saying plainly, because leaving it out would be its own kind of dishonesty. Avocados also carry a meaningful amount of potassium and a plant pigment called lutein, which concentrates in the retina at the back of your eye. Both are real. Both are worth knowing about on their own terms. But they work through entirely different pathways than the one we've spent this whole time tracing, and folding them in here would mean bouncing between 4 or 5 different mechanisms instead of understanding one properly. Today was about the break. That's the one I wanted you to actually understand, all the way down to the neuron. So, practically, what do you actually do with all of this? First, pay attention to quantity, not enthusiasm. The mechanism we just walked through doesn't require a whole avocado piled onto every breakfast. Something in the range of 1/3 to half of a medium avocado, roughly 50 to 70 grams, is enough fat and fiber to meaningfully slow gastric emptying and trigger sick and pie release. Beyond that, you're mostly adding calories without adding much extra break effect. The reflex doesn't keep scaling forever. You're aiming for a feeling of steady, comfortable fullness, not heaviness. If you feel weighed down rather than settled 20 minutes later, you've overshot the amount your gut actually needed. As a rough guide for what the mechanism should actually feel like, mild, easy satisfaction out to about the 2-hour mark, with hunger returning gradually rather than all at once somewhere past 3 hours. If you're ravenous again within an hour, the fat and fiber in that meal were almost certainly overwhelmed by the carbohydrate portion sitting next to them. Second, pair it with protein, not just carbohydrate. The CCK-releasing cells in your duodenum respond to both fat and the breakdown products of protein, and the two amplify each other's signal. Avocado on top of eggs, or alongside plain yogurt, produces a stronger combined satiety response than the same avocado spread on refined white toast alone. The underlying mechanism doesn't change, but pairing fat with protein gives the ileal break more to actually respond to, rather than asking fat to carry the entire signal by itself. Even something as simple as a boiled egg or a small serving of cottage cheese alongside your avocado is enough to add that second signal. You don't need a large portion of protein, just its presence in the same meal. Third, time it ahead of your busiest stretch of morning, not after it. Given that the slowdown in gastric emptying and the steadier glucose release that follows typically lasts somewhere in the range of 2 to 3 hours, eating this kind of breakfast right before meetings, errands, or physical activity puts that stability exactly where you'll use it, rather than burning it off during a quiet, unhurried hour with nothing scheduled. And fourth, the honest caveat, not a footnote to skim past. This will not detoxify anything. It will not meaningfully speed up your metabolism. It will not, by itself, produce weight loss. What it does is nudge two real, mechanical processes: how fast your stomach empties, and how clearly your gut talks to your brain about being full. That is a genuine, usable lever. It is not a solution to appetite or blood sugar on its own, and treating it as one only sets you up for disappointment on the days your plate still has white bread, sugar, and stress sitting on it, doing exactly what white bread, sugar, and stress have always done. It's worth asking, for a moment, why a system like this would even exist in the first place. A body that evolved somewhere it couldn't count on its next meal arriving on schedule doesn't benefit from processing a rare, energy-dense food in a hurry. It benefits from stretching that food out, extracting every available calorie carefully, and using the presence of that fat as a genuine, physical signal that eating can safely pause for a while. The break isn't a flaw or an inconvenience bolted onto digestion. It's the design working exactly as intended, built for a world with far less certainty about breakfast, lunch, and dinner than the one sitting in your kitchen this morning. So go back to that kitchen. 6:47 in the morning, knife through the skin of the avocado. You already know, now, that what happens next was never really about the fruit at all. It's a valve at the base of your stomach reading what's coming. A set of cells in your intestinal wall releasing hormones with names like seek and pi. A nerve running the length of your torso, carrying that news straight up to your brainstem. Two small populations of neurons, deep inside your skull, quietly arguing over whether you're actually finished eating. All of that, every single morning, humming underneath a moment so ordinary you'd never normally think to look at it twice. Maybe that's the real thing worth carrying out of this. We spend so much energy chasing dramatic interventions, cleanses, detoxes, some new pill promising a shortcut, while the body has been running a quietly engineered braking system the entire time, for free, simply waiting for us to feed it something it was built to slow down for. Sometimes the most sophisticated thing you can do for yourself isn't found in something new at all. It's found in finally understanding something that was sitting right there on the plate the whole time. A small, quiet machine that was never asking for your attention, only for you to stop rushing past it.